our next speaker to the podium, Michael Widjaja, and he will be talking about breaking the wall of bacterial defenses.
Thank you. I want you all to try this exercise with me. Take a deep breath. Now only exhale 10%. Now inhale and exhale that 10% again and again. Feels scary, doesn't it? Imagine having to breathe like this constantly for weeks on end. Well, this is what it's like when you have pneumonia. Including influenza, pneumonia is the 12th biggest killer in Australia, costing a total of $84 million in doctor consultations and hospital visits alone. I work on a bacteria that is one of the main causes of pneumonia, and this bacteria is called Mycoplasma moniae. Due to the size and structure of this bacteria, we're limited in the number of antibiotics we can use to treat an infection, and this results in a prolonged disease state. Within Mycoplasma, I'm looking at its proteins. So like other bacteria, Mycoplasma use these proteins to attack and use these proteins to defend, much like the spear and shield you see on this little guy here. Now, if I were to take these weapons and break them in half, they would be useless. But somehow bacteria have evolved a way to actually use these broken pieces through a mechanism called cleavage. So in essence, instead of one spear and one shield, the bacteria can cleave these weapons and will now have two of each. And this essentially increases the number of weapons it can use to cause disease. So what I've found is that Mycoplasma moniae has cleavage, and this has never been reported before. In the lab, I infect human cells with the mycoplasma, and I'm also trying to identify what are these weapons and what are these proteins that are created from cleavage. My ultimate aim is to figure out how this cleavage works and then develop a vaccine against Mycoplasma moniae. This vaccine will give our body the secret to stop bacterial cleavage and thus break one of the walls of bacterial defenses. To achieve this aim, I need to figure out how many of these cleavage proteins there are, what are these proteins, and what do they do. What I've found is that Mycoplasma moniae has so many more weapons than we expected, and because of cleavage, this number just keeps on growing. So my theory is that because it has all these extra weapons, it can easily repel the attack that our immune system launches. And it's this battle, right, that causes this prolonged disease state of pneumonia. At my institute, we have an established pipeline to test this theory, and I've already recreated one of these cleavage weapons in the lab. And I've shown that this cleavage weapon can target the human lung cells. My next step is to recreate other cleavage weapons and see how they interact and how they can be used as defense against our immune cells. And so if we can figure out these proteins, we can break them and make them useless before the bacteria can actually use them as weapons. And so if we can block cleavage, we can breathe a sigh of relief knowing that we've broken one of the walls of bacterial defenses. Thank you.
Thank you, Michael. Questions, comments, right here in the middle, please.
I think you mentioned in your presentation, one of your questions was how many of these cleavage proteins are there? And I think you mentioned you've identified one. Do you have a thesis or a hypothesis about how many there are? Are we talking tens, hundreds, thousands?
So generally the number of weapons that this bacteria has is about 600 proteins, and I've found that about 150 of these have that cleavage event. So essentially you can multiply to make 150 more weapons if needed.
Question here on my left.
So how scalable is it across different types of bacterial infection?
So in my lab at UTS, we're looking at these cleavage events in other types of bacteria. So we found them in a pig pathogen and also another human pathogen, Staphylococcus aureus. And we do have the tools to actually look for these cleavage events. So, but so far what we've searched for, we've found these cleavage events in these different types of bacteria. Yeah.
Great. Thank you very much for the discussion and the answers. And let's thank Michael again.